Hello, my name's Aaron Oakely and I am an Associate Professor here at the University of Wollongong. And today I'm just going to tell you a little bit about a hobby of mine, which is making three-dimensional models of protein molecules and other kinds of biological molecules. So I've been a structural biologist for about 20 years. As a structural biologist, I'm interested in things like proteins and nucleic acids. And just recently I've got into 3D printing, so using fairly standard 3D printers we're able to make these lovely models. The one I'm holding right now, this in blue is a nucleosome core complex, so the histone, is in blue, and this orange stuff around the outside is DNA, and the DNA wraps around the histones, and in eukaryotes a lot of our DNA is packaged in this way. Over here we have, this is a retinol binding protein. The backbone of the protein is sort of in what we call cartoon form. And in orange, you might be able to see inside, is retinol. What else? Over here, okay, this is sort of a classic protein for us. This is myoglobin. It's the first protein structure ever solved. And myoglobin binds to heme. Heme is that red thing in there. And the heme binds to oxygen. So this is an oxygen storing molecule. This is quite a nice looking protein. This is a leucine-rich repeat protein, and you can see we have this sort of repeating motif that spirals around. So some of the structures are quite beautiful, I think. Recently we published a paper where we demonstrated that some non-steroidal anti-inflammatory drugs have antibacterial effects. And what we found was that these particular non-steroidal anti-inflammatories inhibit this protein. They bind to this protein. This is called a DNA clamp. As you can see, it's a donut-shaped protein. And its job is to slide along DNA and recruit other proteins that are important for DNA replication and repair and keep them close to the DNA. And basically, these non-steroidal anti-inflammatories block the interaction of this with the partner proteins. And I have another model here. This is not from bacteria, but it's an analogous system to what happens in bacteria. You have these clamps, and to be placed onto the DNA, they need to be opened up. And you have these clamp loaders, which come along. Clamp loaders bind to these clamps, open them up, and load them onto DNA. So in red, you can see there's some DNA bound in the center, and around the outside is this clamp loader complex. There's a key interaction. Again, this is not from bacteria, but bacteria have the same system. There's a key interaction, which is where the white and the purple meet, that allows this and this to interact. So in the bacterial system, it's the inhibition of this kind of interaction that is the source of the antibacterial activity.